morning, everybody. I guess it's still morning. So it's such a pleasure for me to introduce Andrew Davies here. And so in coming to Cary, besides the delight of getting to work with many of you, it was really fun to come to this area and think about the labs that are nearby that I have synergies with. And Andrew is certainly one of those. And so Andrew is currently at Harvard University as associate professor. Before that, he was at Arizona and at Santa Barbara with Greg Asner for postdocs. And then before that, he did all of his degree work in Pretoria. During that time, he worked with a good friend, Kate Parr, which is where I started hearing about and learning about termites. So we have some interesting ways that we introduce one another and different people so that we're all now working on termites together. And so in Andrew's work, he is thinking about how animals interact with one another and their environment to affect ecosystem processes, and he layers in conservation in that. And I think lots of people love, say, elephants or think about the behavior, but I think what's really interesting is this dearth of people studying how animals affect biogeochemical cycles. And so it's really exciting to have Andrew as one of those labs that's really pioneering that. And so I think the first time we started talking was because he was pioneering interesting ways to use remote sensing to study questions in his system, but we were really wanting to start doing this with Paulo, who just talked and was with us. And so he helped us to pick out our first drones when we were working in Brazil. But then the other thing that's an even greater synergy is he loves termites and works on termites. And so I had the joy of being down in Kruger with him first time we met at the conference that Jane just went to, and standing on a Macrotermes mound and getting him to point, he was pointing out the differences in the grasses on and off of the mounds and starting to think about these processes. So he does cool things like he has a grant to look at the interactions between rhinos and termites and are they facilitating one another. And so you'll hear some of those stories today. But so it's such a pleasure to have him here with us and not so far away from us. And so I hope you enjoy his talk today. So thanks, Andrew.
Thank you. Sorry, I meant to put this on before, but. Thanks, everybody, and thank you so much, Amy, for inviting me and for that kind introduction. And it's been a really wonderful couple of days being at the Cary Institute. It seems like such a wonderful community and place and setting and all of the above. So it's been really wonderful to be here. So thanks very much for the invitation. And like Amy said, the kind of work I do is thinking about these interactions between animals and landscapes and thinking about the way a landscape looks and functions. What is the role of animals in shaping that? So how do animals shape these ecosystems? And so when we look at, as an ecologist, when we look at a landscape like this or like any others, it's easy to think, well, why does the landscape look the way it does? Why are there trees where they are? Why are there water bodies there? Why is there topography? And we can map a lot of this, and we can see the human impact, for example, in these roads or in these dams. But what you really don't see is any of the animals. You can look at this landscape and you can't see any animals, and so sometimes it's difficult to think about what the role of animals is in shaping these landscapes. And this is from Tuklirian Mfolozi Park in South Africa, which is one of the highest densities of animals or ungulates on the planet. And yet you don't see any animals in that image, but they are there. So how do we measure their impact? And that's the kind of work that I do. And when we think about animals, we all know that they have massive impacts on these ecosystems. We can think about herbivores, in this case wildebeest in the Serengeti, and the amount of biomass that they consume and turn over. Or we can think of trophic cascades and predators and how they shape the trophic cascades through these ecosystems. We can also think about ecosystem engineers, whether it's large elephants knocking over trees or powerful termites concentrating nutrients in mounds. We know that they have these impacts, and we know that they do profound things to ecosystems. But when it comes to these global cycles of various things, and this is the carbon cycle as one example, you'll notice that there's a lot of arrows going up and down showing us where carbon is going. There's volcanoes feature here, there's fires, there's industry, but there's hardly any animals in this picture. In this one, many of them don't have any animals at all. This one has some animals in the picture, at least, but you notice a couple things. There's no arrows going back and forth from the animals, and also the animals are livestock. And so there's some consideration maybe of livestock, but when it comes to wild animals, there's very little consideration given to the role that they play. It's somewhat considered negligible in terms of the biomass of vegetation relative to animals and things like that. But when you think about the things that they do, in this case, sticking with the carbon example, this is a review that we wrote a few years ago, thinking about how animals shape different carbon pathways. And the point is that you can see there's lots of ways that they do that, through trampling, through herbivory, through death, various ways that they shape these carbon pathways, and yet we don't consider them when it comes to these big global models. Another example is this is some work we've done on hippos, and thinking of the way that hippos shape aquatic and terrestrial environments. And you'll notice that they do a lot of things. They create these grazing lawns away from the river. They bring nutrients into the river through nutrient transport that increases biodiversity. And when you have hippos absent from the system, you get fires coming a lot closer, lower levels of biodiversity. And this is just one species. So if one species of animal is having such large ecosystem level impacts, what is the sum of all of these impacts on these ecosystems, and how do we measure those? Another important thing is that not only are we not adequately considering the role of animals, but animal communities are changing very rapidly. And this is a scene that you may picture of when you think of an African savanna. You may think of these beautiful scenes of lots of wildlife, but in reality, those places are disappearing, and we're getting large places where wildlife are going extinct. And not only are they going extinct, but we're losing these big species first and getting these very different assemblages of different smaller bodied herbivores. In other places, livestock are being replaced by livestock or being mixed with livestock, and so we're changing these communities all of the time. And then occasionally, there's these bright hopes of restoration where we have rewilding and animals coming back. And this is a composite image from Gorongosa National Park in Mozambique, where we do some work. This is not all at one moment. This is a composite image over about 24 hours, but we get these places where wildlife are being restored. And so the real question is, this all leads to some important questions when we think about landscapes like this one, which is in Kruger National Park in South Africa. And the kinds of questions we ask is, how do animal communities shape these ecosystems? What are they actually doing? And what happens when these animal communities change? And importantly, how do we measure these effects? And I think this last point is important because a lot of the reason for them being negligible is perhaps not that we don't think that animals play a role, it's just we struggle to measure these roles at large scales. We can't see them in satellite imagery, for example. And all of this is coming at the time when there's huge advancement into the way that we study animals. And these are some examples of GPS devices that have been put on animals, and we're in this golden age of biotelemetry where we can put collars on everything from insects up to elephants and everything in between. And yet when it comes to this great technology, we often pair it with relatively coarse remote sensing, essentially. And this is an example of white storks in Europe. So each of these black dots represent a location of a white stork. And you can imagine it must be pretty challenging to catch a white stork. I've never tried it. And to put a device on the back of a bird that flies. And yet, when we think about what are these storks doing, we have this Lancet imagery, which is relatively high resolution, 30 meters, but at the scale of a stork, they're interacting at much finer scales. And so you can see a little bit that a bunch of them are there in a forest, but you know nothing about the forest. Some of them over here on some agricultural land probably, but we have no idea what that agriculture is. And so we can't really see the world the way the animals do. And so what we need to do, if we want to understand how animals interacting with the planet or shaping the planet, we really need to see it the way that they see it and at the same sort of resolutions of the animal data. And so what we do in our lab is we try and fill this gap using high resolution remote sensing, which we use on drones. Drones as well as aircraft. And this is an example of one of our drone platforms flying in the Republic of Congo, which we'll talk about a bit later. And we have the sensor system that we connect to these various drones. And through it, we collect primarily 3 streams of data. So we do LiDAR, RGB, and thermal. We use these together to map these landscapes at very high resolution. So over here, you can see, for example, the shade under these trees. We can use it to measure the RGB to measure dead trees, for example, and then the LiDAR, which we'll get into a little bit later, or the next slide actually, to really think about the structure of these ecosystems. Now, I'm sure most of you are quite familiar with LiDAR nowadays. It's becoming quite popular, but in case you're not, the way it works is that you have a sensor, which could be on an aeroplane or a drone or a terrestrial scanner, whatever it may be, and it sends a pulse of light down to the surface, which then interacts with the surface and bounces back to the sensor. And because we know where the sensor was when the light left, we know the speed of light, we can reconstruct these landscapes in what we call point clouds. And this is a point cloud example of a tree in a savanna from the sensor that we use. And each of these little dots on the tree represent a place where the light interacted with the surface and went back. And so we can essentially make these 3D point clouds, is what we call them. And when we expand it over a landscape, this is an example from Kruger, we can begin to see the variation in tree structure, for example. Across the landscape, this is tree height. We can also look in this cross-section at the 3D representation in landscape. And we can strip all the vegetation bare and start looking at things like termite mounds in this case, and you can see how they pop up out of the surface of the digital terrain models. And what we do is we then combine these kinds of data with experiments on animals or animal data of various kinds. And this is an example of an exclusion experiment. You can see the fence line over here. Where animals have been excluded for about 20 years in Kruger, where they've had access, we can begin to see the differences between areas with and without animals. And these are just some videos of some point clouds from exclosures in Kruger. Inside and outside, the only difference between these landscapes is a fence. So the only difference is large herbivores have been kept out. And you can see immediately these differences. You don't even need statistics to really understand that there's a difference between these two treatments. And so we combine these kinds of experiments of animal manipulations with high resolution remote sensing to get at the role that animals play in shaping these landscapes. And so we don't really view ourselves as a remote sensing or methodological lab, but we do some of that along the way to think about how we can use these data sets to best answer these questions. And this is just one example of how we're thinking about how the elevation of how high you fly affects the vertical profile of the plants that you can measure. So all of these are areas of vertical or graphs of basically fractional cover per 10 centimeter height band of this particular tree. And you can see how you can get various profiles and how they deteriorate with altitude. And this matters because it's always this tension between extent, how much area do we want to cover, versus resolution, how well do we want to measure the things that we measure. And it depends on the kinds of questions, and we'll get to this at the end, how we tie it all together, is thinking about remote sensing to think of broad scale patterns, and then high-resolution data with experiments to think about mechanisms. This really high-resolution data has also allowed methodological advancement in ecological studies. For example, this is an example from Kenya about how ants shape trees. And these are, if any of you have been to Impala Research Center, which I'm sure some of you have, these are Acacia drepanalobium, which have these very tight mutualisms with ant species, that 4 different ant species occupy different trees, and depending on the ant species, the phenotype of the tree is very different. And so you can go and do these surveys, but they take you days to go and survey every tree, survey which ants there are, measure the structure of it. But what we could do is we could use this high-resolution LiDAR data to pull out these point clouds of individual trees. And these are 2 different ant species, Chromatogaster, well, they're both Chromatogaster species, Nigroceps and Mimosiae, that have very different phenotypes in the trees. The trees look very different. And you can go and identify each individual tree yourself, or you could use this high-resolution data to do it in essentially an hour. So we flew the forest geoplot at Impala in about an hour. Obviously, there's a lot more processing time. But we could pull out these point clouds of these individual trees and make maps of which ones are occupied by which of these 4 ant species. And the models are not perfect, but our accuracy was only around 50%, 40%. But what's important is if you compare the field data to the remote sensing classification, it's really similar. And so these are different analyses of field data on the top, and the same analysis using this classifier, this machine learning classifier from the LiDAR data, and you can see that although there's differences, the patterns are essentially identical. And so we can use these kinds of approaches to really scale up our measurements and be far more efficient at them. And so today, I'd like to go through a few examples of how we think about the way that animals shape landscapes and use these different data sets. And we're going to think about how large animals like elephants or tropical forest communities shape savannas and tropical forest landscapes that they inhabit. We're then going to think about abiotic controls and think about how things like fire interact with animals to shape these landscapes. And then lastly, we're going to think a little bit about how we can turn this around and think not only about how animals shape landscapes, but how animals respond or use landscapes that can be useful for conservation. And we'll think about things like African wild dogs, as well as my personal favorite, which are termites. So before we get there, you may have noticed that all of these examples are from Africa. So why do we study Africa? Well, because it's the best continent. And if you don't believe me, you should go visit. But if you want a real scientific reason, it's because Africa is the last continent with these large intact animal communities that haven't been lost. And they used to be present on all of these continents and largely went extinct during the Pleistocene for various debated reasons, but without getting into that, you can see that Africa, in this case for mammals, has the highest diversity. And so we can use Africa as a model system to understand how animals shape these ecosystems and how places like South America may have changed as a result of losing their larger mammal communities. And so the first example I want to think about is thinking about large herbivores and how they impact vegetation structure. And we know that they have direct effects on individual morphology of trees. For example, this tree that has been destroyed by this elephant is never going to be quite the same again. And they have these large effects. We know they also maintain grazing lawns, for example, and affect the herbaceous layer. We know there's a lot of top-down control of tree grass and balance between or driven by herbivores. But what we don't really know is how these direct and indirect effects affect plant diversity and the relationships between diversity and structure. And so a postdoc of mine, Tyler Coverdale, led this review where we looked at a meta-analysis of looking at relationships between vegetation structure and plant diversity. And you notice that all of these are different measures of diversity and different measures of structural complexity, but almost always there's this positive relationship between diversity and structure. And if herbivores are affecting structure, are they affecting this relationship? But over 90% of these studies were comparing diversity and structure from forests. Very few were done in savanna systems or in grassy systems. Very few manipulated diversity, and very few, if any, tested the effects of animals. And so we wanted to understand, how do animals affect these relationships between structure and diversity? And so to do this, we went to Impala Research Center in central Kenya, which has these exclosure experiments set up. This is an example from Google Earth of these 4 different treatments that have these different levels of exclusion. So we have a control with everything. We have then some that we lose only the mega herbivores, only the mega and the meso, and then all of the herbivores. And we were interested in thinking about how exclusion at different body sizes affects the impacts on the communities, because as extinction happens, we generally lose the larger animals first, and the smaller animals stay behind for much longer. And so to give you an example, we combined LiDAR data with field measurements that have been collected by many collaborators there for a very long time. And this is just some example of the different structure of the vegetation from the LiDAR data over these different treatments. And we had this conceptual diagram to think about. What we're trying to think about is how herbivores affect tree and herbaceous diversity and how that works together to affect vegetation structure. So this diversity-structure relationship, and how herbivores also directly affect structure themselves. And so there's a lot of results that came out of this. Some things quite surprising. What you notice is when it comes to herbivore effects on trees, essentially once you lose mega herbivores, the result stays the same. So if we lose the largest herbivores, we have these big effects on tree abundance, as well as diversity and tree density. So it's only really mega herbivores, in this case elephants and giraffe, that have these big effects. When it comes to the herbaceous layer, you'll notice there's some differences. So there's these big differences when you lose mega herbivores, but especially in diversity and abundance, you start getting these big differences when you lose mesoherbivores, which I think you can see most clearly with the abundance. And so the medium and small-bodied herbivores have larger effects on the herbaceous layer than they do on the tree layer. And when we think about how these relate to structure, this is a relationship between tree diversity and structure. And you can see for almost all of them, there's this positive relationship that we would have expected. But also, it saturates very quickly. Once we have from 0 to 1, we actually have very similar relationships, except for percentage cover that kind of increases with diversity. And when it comes to the herbaceous layer, how does herbaceous vegetation affect structure? We see very similar patterns. Essentially, this increase of diversity leading to increased structure, somewhat more linear in some of them than the saturating patterns, but we still get this saturation. So there's some kind of functional redundancy. And then lastly, we wanted to think about, well, what about direct effects of animals on structure? And we found that there's very different responses per tree species. So of 5 dominant tree species in these landscapes, only really 3 of them were affected by herbivory. And again, you see this very strong pattern of once you lose the largest herbivores, that's where you get the biggest changes. But if we were to zoom into one of them, which is Acacia mellifera, you can look at it. We looked at individual trees from the point cloud data that we'd identified in the field. And you'll notice that individual trees, when you don't have herbivores, become a lot smaller in terms of the height as well as their crown area, but their densities don't actually change. And so when we think about how do herbivores shape the structure-diversity relationship, is the first thing is we know that they actually reduce diversity, which is interesting. In many places you would have thought they would have increased it. But generally, we still get these more diverse communities or more structurally complex. So if we can have situations where herbivores at larger scales, because these are pretty small plots, increase structural diversity or complexity, we may see increases in diversity. And really what it comes down to is herbivore extinction will alter this vegetation structure biodiversity relationship, both directly and indirectly. But herbivores don't only change vegetation structure. So this is an example now from Kruger National Park, so a lot further south in Africa, looking at these exclosure experiments like we've seen before and seen these big differences across the fence line. But that's not the only thing they do. They also have these cascading effects on things like thermal landscapes. And so we combined LiDAR data with thermal imagery to look at differences inside and outside of these exclosures on surface temperature using the thermal data. And you can see immediately that inside of these exclosures, there's a lot more of these cooler temperatures all over the landscape versus outside where they're much reduced. And if we're thinking about thermal landscapes and the scales at which it matters, this is an example of a 25-meter macroclimate model from 25-meter satellite data. Now, 25 meters we might think is pretty fine scale, which it is in the scale of the globe. But if you think about how you interact with the thermal landscape on a hot day, do you care about the temperature in this whole room, or do you care about these very select little spots in the landscape? And so this wildebeest here, there's two of them, are selecting for these very fine, very high resolution, fine spatial scale patterns. And so if we want to understand the impacts of microclimates, we have to really look at it at this fine scale. And so when we take the same area here and we use 0.5 meter LiDAR data, we can really begin to map the same area in much higher resolution. And so here, suddenly, things that are somewhat obvious over here really pop out to where these higher temperatures are and these cooler spots are, which are of much more significance to organisms. And so we wanted to understand the interaction between herbivores and thermal landscapes. And so we went to Kruger, and we looked at these exclosure sites that go from the north of the park all the way down to the south. So there's a strong rainfall gradient. There's a geology gradient with granites and basalts, which have implications for soil fertility. And we collected surface temperature across these fence line contrasts, as well as vegetation height. And then we actually looked at plant area index from the LiDAR data to also think about how height and foliage density really affects thermal landscapes. And so we looked at these patterns across the fence lines and the drivers of them. And these are examples or kernel density plots from these 6 exclosure experiments. This is outside and this is inside, and these are basically the kernel density of temperature. And you notice that all of them, temperatures are much lower inside the exclosures than outside, across the rainfall and geology gradients. When we started combining this with the LiDAR data to think about, well, what's the relationship, what's the driver of this, you see these interesting sort of triangular patterns between surface temperature and vegetation height. Where you get really tall vegetation, you consistently get these lower temperatures. But when you have shorter vegetation, you can get this whole range of temperatures. And I think that's due to things like shading. So you can still have short vegetation that's in the shade, which can give you these lower temperatures, but you also get these higher temperatures. So you get consistently, the only places where you get consistently low temperatures are where you get tall vegetation. And looking inside and outside, as I'm sure you'd expect, you can see much taller vegetation inside the exclosures than outside. And when we look at PAI, it's exactly the same kind of pattern. These areas with high PAI or dense canopies are consistently cool. Areas that have sparse, low levels of PAI, so sort of sparse vegetation, you can have this kind of mix. And again, obviously, we get these much higher levels of PAI inside than outside these exclosures. And so when we're thinking about the drivers of this, of course, there's a lot of meteorological variability, and it could be what's the temperature on the day, what's the conditions on the day. So we built these models, some GAN models that included all of these meteorological data, and found that across all of these exclosure sites, these are the sites, we get this very strong relationship between the effect size of height and temperature as well as PAI, and Cohen's d is really a measure of effect size. And what you're thinking about here is this slope is showing that when you have high Cohen's d in height or PAI, you get very high, similarly high, in this case negative, but strong effects of Cohen's d, which basically means the negative Cohen's d for temperature indicates that there's lower temperatures inside these closures than outside, and they're driven by these vegetation structural differences rather than climate or meteorology. And what is really interesting is when we divide this into woody vegetation and grass, which we can do with the data, at the resolution that we work with, we can separate the grass layer from the tree layer and really measure the grasses. You notice that the difference in temperature at woody vegetation inside and outside is really not that different, where the trees inside or outside is going to be quite cool. But when we look at grasses, we can see there's this really big distinction between the surface temperature of grasses inside and outside. And this comes back to those triangular shaped patterns over here, where you can think about grasses being in this sort of level that can be really different. And when it comes to the grass area, that's where we see these differences. And that's of consequence because as we change these wild communities of wild animals with livestock, we're essentially replacing a browser-dominated community with a grazer-dominated community. The number one livestock species, especially in Africa, is cattle and sheep, which are grazers, whereas the predominant herbivores are things like elephants, which are mixed feeders, but predominantly browsers, as well as giraffe and kudu and these kinds of things. And so when we think about how animals affect these thermal landscapes, there's been a lot of emphasis on how microclimates are going to affect animals and organisms, but we kind of show with this work that animals themselves can drive changes in these thermal properties. They're not just passive bystanders, but they're active players in this process. And this will have ecosystem level implications, especially as we change wildlife populations and think about the replacement of wildlife with livestock that are going to have these stronger effects on the grass layer. So with that, we're going to now change gears and move to a completely different place and move from tropical savannas into tropical rainforests, where we work in the Congo Basin in a national park called Ozala Kukua National Park in the Republic of Congo. And there we're studying these huge clearings that are called bais in the local language. They're the world's largest natural clearings, but we know very little about them. We don't know how many there are, we don't know how they formed, we don't know if they're important, and what we also don't know is the role of animals in shaping them. And what you probably didn't notice is that there's a whole lot of forest elephants along the edge of this bai. And these bais are hotspots of animal activity, and do these animals actually play a role in shaping them? And so the first thing we did is went to the site, and there it is up there in the Northern Republic of Congo, and mapped where the bais are. So we used LiDAR data to build a classifier that then trained satellite data to map the spatial patterning of these bais. And this is a map of the park, and all of these red areas here represent bais that we could find. There's a bit of a zoomed in area over there. And we found more than 2,000, whereas the park before had thought that there were only about 200 of these bais. And so a lot of them had been completely undetected. And so we then thought, well, how are they distributed across this landscape? And this is a Ripley's K diagram, which this black or this red dotted line represents what a random distribution would look like. Anything below the line would be even spacing or over dispersion. Anything above the line would be indicative of clustering. And so you can see across all spatial scales, these bars are clustered. So they're in these specific places. And this is an example of a cluster of these bars, and you can kind of see it on this map too. And so we learned that they're clustered, they're kind of near rivers, they're in these flat areas, there's some kind of hydrological signature. There's lots of them, over 2,000, but they're not everywhere. In fact, they only represent about 1% of the park area. And then we wanted to ask, well, are they important? Do they matter? And this is a great example too of some indigenous knowledge that kind of inspired our work, because whenever we went there, we'd map these bars and they all kind of looked the same to me. But all these local people would tell us, well, there's elephant bars and there's gorilla bars, and they're really different. And I was like, oh, they're really different. But we decided to test their hypothesis, and we looked at these different types of bars and we surveyed plant communities within them. And what we find is, this is an NMDS of the plant communities. So these blue and purple areas represent the communities of the different bars. And you'll notice that these elephant bars are completely different to these gorilla bars. In fact, these are dry and Dembe savannas. The savannas are more similar to the bars and yet very different than the bars are from each other. And so they're extremely important for plant diversity, and in fact have different communities that are also different to the savannas. So they're not these remnant savanna patches. And so we realized that these bars are important for biodiversity. We realize that they're aggregated, but they're really small. But what we still don't know is why are they there? How are they maintained? What we do know is, like I said earlier, that animals love them. And these are pictures of elephants and gorillas and forest buffalo and Sitatunga that really congregate on these bars, often focus their home ranges on them. If you look at some GPS data from elephants across this region, you can see all of these concentrated areas where elephants spend a lot of time are generally concentrated on these bars. And what they're doing here is they're mining the soil. This is some example of some trampling around a mining site. They can even dig out underneath the trees. And it seems that they're here to get it salt. These are sodium-rich sites. And so we know that these animals are coming to these bars. They also come for social reasons. There's a lot of open space. There's a lot of various reasons. Perhaps it's for salt, perhaps it's for social behavior. But what we don't know whether, why exactly they're coming and what their role in shaping them is. So we have this idea that it's the salt input. So what we're doing is we're taking 6 of these bars, this is one example, and we're adding tons of salt to about 6 of them and leaving another 6 as controls. And this is one of the sites where we put the salt, so we put it in these concentrated areas, we bury it to kind of mimic what would happen. And if you look at this one area here, this is the area that was circled. This is the activity after only about 3 weeks, I think, after the salt had been put in. You can see all of these footprints coming into these areas where the salt was. And so what we're doing is we're combining with this experiment a lot of field surveys to understand how the vegetation is changing. We're taking soil samples, or at least we as in lots of students are doing all the hard work. Collecting soil samples, we've got some bioacoustic monitors up to understand biodiversity within the bars. And then we're combining that with camera traps along the edges to understand and quantify whether an increase in sodium will lead to increased visitation rates by large mammals. And then we're combining this with LiDAR data to map the edges of the bars in really high resolution in a repeated manner over a few years to understand whether the bars are increasing or decreasing with added salt. We're also expanding this to think about how these bars influence ecosystem function. And the way we're doing that is through the lens of termites, essentially. And measuring termite communities in relation to the bars, but also doing decomposition experiments where we're thinking about the role of sodium in decomposition and termite activity. And so what we've done here is we have this factorial experiment where we have different mesh sizes that allow termites in and exclude them. And then we've soaked half of them in sodium or salt water, essentially, and half of them not, to understand what the level of attraction to sodium is for termites. And these data are really preliminary. They've only come in just recently. But we had decomposition in the forest along the edges of the bar, and then also in the savanna. And these are the ones that have salt, that were soaked in salt. And you can see, especially for savannas, there's a huge increase in decomposition for the ones that were soaked in salt. And so we were thinking about the way that this kind of feeds into these ecosystem functions. And all of this is against this backdrop of whether these bars are stable and whether they're disappearing. And this is an image from the DRC in an area where elephants have been excluded for a long time. They were essentially hunted out. And this is the remnants of a bar that is closing in and becoming basically swallowed up by the forest. And so we're trying to ask, what is the value of these bars to the Congo Basin, and what's going to happen if we lose these animal communities? Are they really fundamental in keeping them active? Now, as much as I love animals, and I'm an animal ecologist, I also recognize that it's not only animals that are driving these patterns. And so we also think about abiotic drivers, in this case, fire. And we think about fire because it's got a lot of analogies to herbivores. It really functions as an abiotic herbivore in many ways by consuming biomass. But it also does some very different things and interacts with herbivores. So a lot of studies, including our own, think about how vegetation structure changes with fire regime and fire frequency. So these are examples of experimental burn plots in Kruger National Park. This one hasn't been burnt in about 70 years. This is burnt every 3 years. This is burnt every year. And you can see the differences in the vegetation structure. But the problem with these kinds of approaches is that not all fires are the same. And not a single fire is homogeneous in its effects. But when we look at a plot and we think, well, this plot burnt every 3 years, we're kind of making the assumption that the fires have been the same every year. But they're really not. And the same way that individual animals are not the same, individual fires are not the same. And so we started an experiment down in southern Kruger National Park where we burnt about 40 different patches. Here they are, they're 1 hectare patches with fire. We collected LiDAR data before and after the fire. which we could then use to map grass biomass. So we're looking at the grass layer and understanding how much fuel availability there is, and then combine that with thermal imagery. We'd have a drone above the fire taking an image every 2 seconds. And so here's an example of what that experiment looks like. So we had these plots, 40 of them, and this is after the fire. You can see how fires changed quite a lot and their patterns and their burn scars. And then during the fire, we took these thermal images, so you can see how the fire spreads across these 1 hectare plots. And then we combined these thermal data with LiDAR data from before and after. This is before the fire, and this is an image of the LiDAR data after the fire, looking at the grass height. These are very grassy systems, as you can see in the video. Trees are not very common here. And we're combining these data sets to understand how changes in fire intensity relate to changes in biomass consumption. And what we find is, firstly, we burnt these fires also at different times of the day to kind of mimic different intensities. These are 3 different measures of fire intensity that we derive from the thermal imagery: residence time, rate of spread, and the maximum slope of temperature, so how quickly they get hot, in this case above 60 degrees, which is what's physiologically important for plants. And you notice that there's a lot of variability in these fires. Not one fire is the same. And some of them, like early morning fires, are quite homogeneous. Others are really quite varied within the fire themselves, and there's very much variation between them. And when we compare that to what it does to vegetation biomass change, you can see similar variation in how these fires are interacting with the grass layer. And what's interesting too is some fires that are quite homogeneous in their intensity still have variable effects on the grass layer, whereas others that are quite homogeneous have homogeneous effects, and that are variable have variable effects. There's a lot of interactions here between variability of fire intensity at fine spatial scales and vegetation change. And when we think about what that means, is these are different, again, the different levels of fire intensity and the absolute change in biomass. And these are early dry season fires and late dry season fires that are spread across these times of day. And you notice that as you increase intensity, you increase the amount of biomass that's consumed. But what's interesting here is that there's a lot of sort of pressure to move fires into to do more early season burning, because that reduces emissions. But you can see here that actually the amount of absolute biomass consumed by these early season fires is often higher, and that's because there's more stuff to burn. By the end of the dry season, the grass is drier, the fires are more intense, but there's less to burn. When we think about it as proportion of biomass loss, so this is the same plots, but instead of absolute biomass as proportion, we can see that a higher proportion of the biomass is burnt in these late dry seasons fires, but actually more biomass is consumed in the early dry season. So these are very different implications for emissions, as well as for habitat heterogeneity and ecology, because a fire that burns most of the biomass, in this case like 75% of it, will have much different implications for heterogeneity, biodiversity than a patchy fire, but also different things for emissions. But now we still are animal ecologists, and so what we really want to do is think about how fire interacts with animals. You know, what happens when these 2 different kinds of herbivores interact? And fire interacts with herbivores in lots of different ways. This post-green flush that comes after a fire is very attractive to animals, and they forage on it, and this can also cause these grazing lawns to occur where animals are feeding and concentrating their feeding on this post-fire green flush. But if the fires are spread out too widely and really large, this concentrating effect doesn't really happen. And so also in Kruger, this was Central Kruger and Satara, we started an experiment where we looked at different sizes of fire, so these were in different seasons, and we went from everything from a 1/4 hectare to 25 hectare fires, burnt them for 7 years, and then turned them off. And so what happens if you burn these fires, you create these short grass patches that bring in the animals, and then you stop the fire. Can the herbivores actually take over from the fire and keep these patches open and as grazing lawns? And then we use LiDAR data. So this is a year after the fires are stopped, so we'd had one full growing season without any fire, and measured the grassland. And you can see in the unburned plots, in these control plots, these pink ones, you get these really tall grasses, but in the early burns and the late burns, the grasses are still short, even more than a year after these fires. And here's just an example of some of these plots looking at grass height from LiDAR data, and really showing how not only are the patches maintained, but they're actually increasing in size. And the herbivores are actually expanding the size of these patches. If we look at some of the data, you can see how this is grass height with the unburned, and the two seasons really have these short grass patches, even when we stop fire. But what you also notice in that plot too, and even these, the size of the fire doesn't really matter, and nor does the season. Essentially, you just need to concentrate this grazing. But interestingly, on early burn fires, you also get these larger patches or larger areas of these shortgrass patches. And these shortgrass patches matter because they're a different kind of habitat, they increase biodiversity. And again, thinking about this push to get early dry season fires versus late dry season fires, we have to understand the differences between them and the different things that they do. So putting this together and thinking about how these small-scale fires interact with herbivores, this is a schematic that we put together. Essentially, if you have no fire, the grasses will senesce, and that's all that will really happen. You get these tall grass specialists like African buffalo in this case. But when you have fires that are really large, you'll get these short grass patches, and things like wildebeest, which are short grass specialists, will come, they'll forage, but they'll be so spread out across the landscape that you don't get any kind of feedback, and a year later it's tall again and you get these tall grass specialists. But when you have these small patchy fires, what happens is you're concentrating these short grass specialists on these small little areas that then create these feedback loops that even when you stop burning, you get these patches that persist in the landscape. And when you do that in an early season, so this is an earlier fire, there's more time essentially for these feedbacks to develop before the rains come and before the grasses grow, and you get these even larger patches and these feedbacks between herbivores and fire that can actually have these long-lasting effects on these landscapes. And so the last thing we want to talk about is thinking about how we can use these kinds of approaches to understand how animals are responding to changing landscapes. And we're going to talk about wild dogs and termites, which are very different, but you'll see how they come together. But when we think about wild dogs, the first thing you have to know about wild dogs is that they're endangered, and also that they don't get along well with lions. Lions will kill wild dogs at any opportunity. In fact, it's the highest cause of wild dog mortality. And so in most places, wild dogs simply avoid lions. So this is an example of the Serengeti National Park. This is this really well-studied lion area that Craig Pack has been studying for decades. And these are GPS points of collared wild dogs, and you notice that there's hardly any overlap. The wild dogs just simply avoid lions. But when you get to the present day situation where you have landscapes that are fragmented and far smaller, wild dogs don't have that luxury. And so this is the Tlhotliwa Game Reserve in northeastern South Africa. It's about 1,000 square kilometers. It's got the highest wild dog density in Africa, one of the highest lion densities in Africa. Wild dogs should not be able to live in this area. And when you look at it over a human population map, this is KwaZulu-Natal, the province, which is where it's located. It's one of the highest densely populated areas in South Africa. And you'll notice that this is the park here, this low human population, but it's completely surrounded by high human population. And so it's completely fenced. And like I said, it has the highest wild dog density in Africa, one of the highest lion densities, completely fenced, increasing human populations. It doesn't make sense that wild dog populations should exist, especially not at the densities that they do. But something else you need to know about this landscape is it's extremely heterogeneous. It's one of the most heterogeneous African savanna landscapes you could find. It's got these patches of dense forest. It's got lots of topography, open grasslands. And so our hypothesis was that perhaps wild dogs can actually use this variability in landscapes to avoid detection by lions. And so what we did is we had collars on lions as well as on wild dogs, and this is a map of where we had wild dog data against this backdrop of lion density. And we did some integrated step selection analysis to think about where wild dogs are moving in this black line versus where they could have moved in relation to lions. And then tested these different hypotheses of whether wild dogs are simply avoiding lions spatially, or whether they're actually using the landscape so that when lions are close by, they actually use it differently to escape detection by lions. And we ran a bunch of models and different analyses and found that overwhelmingly, wild dogs are not avoiding lions, they're actually changing their use of the landscape. And so then what we did is we went to this is part of the process, I guess, is we went to each wild dog site and we measured what we call the view shed using the LiDAR data. So what can a wild dog see? So if you could imagine there's a wild dog here, anything in light colors is what the animal can see, whether it's on the sort of slope or in the vegetation. Anything that's darker is something that's occluded from view. And so over here, for example, there's some topography that the wild dog can't see there. And you'll notice that what's nice about this is you can see that it can see here, it can't see over there, but then when the slope gets higher, it can see there. So you can really measure what an animal can see. And we then looked at how lion encounter risk varies against probability of use at these different view sheds. And so the 10th percentile is a very dense, small area. The 90th percentile is a very open area. And during the day, this is during the day, you'll see that as lion encounter increases, wild dogs kind of use more open areas, but when it's at nighttime, when lions are really active, you get this very strong relationship between wild dogs and view shed, and they're selecting these open areas when they're in areas that are high risk. They're not avoiding the lions, they're just changing the way that they use the landscape. And this is partly because we think wild dogs see very poorly at night, lions see a lot better, and so you'd rather see the enemy coming than try and hide away from them and not notice them. And so when we think about wild dog-lion coexistence or carnivore coexistence, we have to first recognize that wild dogs evolved with lions. So it's unrealistic, really, or unlikely to have natural populations in the absence of lions. So instead, spatial heterogeneity could really provide this alternative mechanism of carnivore coexistence. And we normally think of carnivore coexistence through dietary niche separation, things like that, but this increased heterogeneity could actually be driving these patterns. And when we think of other areas, this is Etosha National Park in Namibia, where it's very open, very homogeneous. That's Etosha Pan in the background. Wild dogs have been reintroduced to Etosha on multiple occasions and every time go extinct. And the same has happened in the Serengeti, where it's really open. And so we should actually be focusing our efforts in areas that are heterogeneous, that can provide these areas for wild dogs to escape lions. And so this is an example of how animals actually use landscapes in a conservation setting. And then the last example, saving the best for last really, is termites, which I've actually done a lot of work on, and this is a paper that came out recently that Amy was involved in as well, thinking about how termites influence methane emissions, which we're starting a project on, but don't have any results yet. But termites are really, really interesting and really ecologically important because of many reasons, but what they really do in these termite mounds is they concentrate nutrients. So they go out there and they forage, they bring the forage in, and they have these high levels of nitrogen and phosphorus as well as moisture, and they create these islands of nutrient and moisture enrichment that changes the vegetation and invertebrate communities on them. And we can map them with LiDAR data, which is really great because it gives us, it's one of the few times that you can actually measure insects with remote sensing, but we can measure their characteristics. And we've done a lot of work to think about how these termite mounds function in natural environments and how they're beautifully spaced like this and what that means for the ecosystems. But most of Africa is not a protected area. Most of it is changing really rapidly due to human population growth and livestock and settlement. And what we wanted to know is what happens to these termite mounds and what happens to the ecological importance of termites. So it's a conservation kind of question. And so what we did is we went to Kruger National Park and measured mound distribution patterns inside the park and then in these private reserves and then outside the park in these communal areas. And these are some examples of communal fields outside the park. This is in landscape 6. And then in comparison with these protected areas, this case in the private reserve, you'll notice that they're extremely different environments. And then we use the LiDAR data to think about how do these termite mounds vary across these different landscapes. Every little red circle represents a termite mound that we used a AR classifier to pull out of. And then we looked at things like termite mound height and density and spatial patterning across these landscapes. And you'll notice that there are big differences. So these green and reds are basically natural environments, and these two are human land use types. And you'll notice that, sure, you get higher densities of mounds in the protected areas and taller mounds. But on average, it's not that the mounds are disappearing. They have about half the mound density as they do in the protected areas. And the height of the mounds, the average height is not really changing, which is important because the height of the mound is related to the size of the colony, which is related to the population and the impacts that it can have. And when we think about the spatial patterning, these are those Ripley's K analyses that I showed you with the bars. These examples over here show that at fine spatial scales, termite mounds are very evenly spaced, which basically means their benefit is distributed equally across the landscape. And then they become clustered at these landscape scales. So they're on these hill crests, and on the hill crests, they are very evenly spaced. And the even spacing is driven by inter-specific competition between the colonies. The clustering is driven by abiotic controls like water inundation and things like that, where they can't be close to streams. When you come to these human areas, you'll notice that it's a lot messier. I mean, look how nice and neat these patterns are across the replicates. But here, they're very varied. But you notice that even in these human landscapes, you can actually see the same kind of pattern persists. And the human landscape, there's agriculture, we kind of lose the clustering a bit, but we keep this even spacing. And if any of you have tried to get termites out of your house before, you'll know how hard that is. And it's the same kind of thing. Trying to get them out of a landscape is actually quite challenging. And what this then means is that when you think about termites and land use, at least half of these mounds remain. There are some big differences in the spatial patterning and the height, which we need to unpack. But because they survive and they're quite resilient to human disturbance, can we actually use these as sites of savanna restoration because of all the benefits that they have? We need to do some more work to understand the effects in the ground, but it's a promising approach to actually use termite mounds in restoration efforts. So when we think about how all of this comes together, these are very different examples, different ecosystems and animal species. The thing we're trying to get at is how do we scale animal effects to landscapes? And I think there's a few takeaways. Firstly, that we can really integrate field and remote sensing data to get at these landscape level patterns. These manipulation experiments like the exclosures, the nutrient additions, things like that are fundamentally important to uncover the mechanisms that we use to understand what the impacts are, but we can use the remote sensing to look at these broad scale patterns. And when we put these together, we can start to understand the interplay between biotic and abiotic drivers of landscapes, thinking about fire and how it interacts with herbivores. And eventually use all these kinds of insights to demonstrate the importance of animals in landscape structure and evolution, to motivate the inclusion in global cycles like the methane cycle or the carbon cycle, and also use this kind of information to inform conservation efforts. And so where all this is going is these stars here represent areas that we've collected LiDAR data, we've combined with animal data across Africa. These hashed ones are areas where we're kind of going to, and the idea is to build up this data library of these high-resolution data sets that are then repeated every few years to look at things like the carbon change. And this is some work that I didn't talk about today, but it's how elephants drive carbon change in Kruger, thinking about these are LiDAR surveys 6 years apart, which we related to a whole lot of different drivers, and to begin to think about how animals are shaping things like carbon turnover in landscapes. And then ideally, once we've sort of done this kind of work in Africa, thinking about why we study Africa, to then translate it to other continents and think about South America, Australia, and think about what's happened to those savannas or tropical forests as we've lost these large herbivore communities or large animal communities. And so with that, there's a lot of organizations that we work with that I want to acknowledge, a lot of funders that fund this kind of work, which we're obviously very grateful for. And then a lot of different people in my lab that do a lot of the hard work and a lot of projects that we haven't had time to talk about, like rhinos and lions and various other things. So thank you.
So questions?
Thank you for your talk. My question is, when you're looking at the differences in termite colonies between areas that have a lot of human influence and areas that don't have, is there a way to know if these colonies are still active? Because I know, like, you can find colonies that are still like there, but they don't have animals inside, and if that can like help to predict better, what's going to happen.
So the question for people in the video is that it's all getting well mapping the termite mounds, but how do you know how many of them have active colonies, basically? Like, can we know whether termites are present or whether these are just relic structures? Because that happens. Termite mounds can last thousands of years, we think, but it doesn't mean that the colonies are active. It's active all the time. And so the only way to do that is really combining it with field surveys. We've thought of a few ways of looking at thermal data to think about whether active termite mounds are hotter, or at least more consistent. And so we've tried that with some thermal data. It hasn't really proved successful yet because they're often covered in vegetation, and that sort of dampens the thermal signature. But we have done field surveys and found, at least in Kruger, about 50% of the mounds are active. We need to extend that kind of work to look at the effects on the ground. That's kind of what I was getting at, is how many of them are active? And what does it mean if we lose half of them? What does it actually mean for ecosystem functioning? So that's kind of the next step on that. But the other thing I will say is the termite mounds themselves are often the thing that really has the ecological impact, because that's where the nutrients are concentrated. And how long, once a mound becomes abandoned, how long does that signature stay? We don't know. Like, what's the longevity of that effect? So we also need to understand, if we lose the colony but keep the mound, how long does that effect last? And these mounds are often re-inherited by another colony. So if you give them enough time, they'll get re-inherited and they'll start being functional again. And so these are all kinds of questions that we need to ask.
Thank you for a great talk. So I know you mentioned that we don't totally understand these buys and how they get formed, but could you maybe mention like what some of the thoughts are and whether it's like geologic or hydrologic, and then how the fact that they're concentrating and then creating hotspots and nutrient cycling might maybe change those control or enhance them?
Yes, the question is really about what forms bias, essentially, and like what do we know, which is very little. But we do know, like, there seems to be a very strong hydrological signature. They're always down in these low areas. They always get flooded in the wet season. So there's something hydrological going on for sure. They're always closer to these rivers. There's been suggestions that they're in areas of dolomite. We don't really have good geology maps for this region, so we can't really test that yet. And then they have these high sodium levels. And so I sort of think that it's this three-way interaction between nutrients, hydrology, at least, and animals. And if you lose one of those three, you will lose the buy, because obviously if the hydrology changes, I think the flooding is important to keep the trees down. If you lose the nutrients, you probably lose the animals, unless they're coming for different reasons. And if you lose the animals, at least things like the elephants, they will probably close in, which is what we see in the DRC. And so I think it's this three-way interaction. There's also been suggestions that humans have started them. There's some pottery and things like that. I don't really think so, just given the number of them and how these human populations have always been very low in this region, but it's one of the lowest human populations on the planet. And a lot of the people have been moved out of those areas and gone to roads and things like that, and yet the bars are still there. So I don't really think it's that. But we need to do some more testing. I'd love to build some exclosures in them, see what happens when you keep animals out. Do the trees come back, or are they kept out by the flooding? We want to take some soil cores and try and understand how old they are and things like that. So there's a lot of questions to ask, but that's kind of what I think it is. I think it's hydrology, nutrients, and animals. Evan?
Really, really cool talk. I have a follow-up related to the exposure effects on plant height that then have the knock-on on temperature and microclimate patterns. Sort of had a two-part. I might have missed this, but I wasn't sure. You showed that within grassland probability density associated with temperature seemed to be a bigger effect than within trees, but I wasn't sure about their relative area effect in the total area in those sites. And in terms of mechanism, have you all explored that at all? If this is related to transpiration or water cycling in some way?
So is your first question about the relative cover of trees and grass in these areas? So the first question is about the relative abundance of trees and grass, and then the second question is about the mechanisms. And so the trees, we could measure the abundance. It's probably, it depends how you sort of define it, because there's often grass underneath trees, for example. But if you were to look at, woody cover generally varies between about 10 and 60, 70%. In the full exposure is a lot higher. It could get up to 80, 90%, but generally outside is probably around 30, 40% tree cover. So majority grass, really, at least in the open areas that are accessible to herbivores. So it's quite large. And I think savannas in general sort of range between 10 and 70%. And then the mechanisms that we haven't done any of that. That's kind of the future work that we want to do. The only mechanism we've looked at is the effect of herbivores on structure and structure on temperature. But what's actually driving these differences, what it means for tree or plant physiology, we haven't got to yet. I think there's a lot more to kind of explore.
So I have a question online. Miriam Johnston wants to know, what does it take to exclude mega herbivores like elephants from a particular area?
A lot of work. It takes, those are electrified fences, the ones in Kruger, so electricity really works. The ones in Kenya too. Anything else doesn't really work. We're building some rhino exclosures too, which are actually easier because you just rhinos can't jump, nor can hippos. So we can exclude those by, about 50 to 70 centimeter long just cable. And we're basically just relying on the fact that they're not intelligent enough to realize they can destroy a cable, which so far has proved right, and they can't step over it. But when it comes to elephants, the only real way to do it is to use electricity. And so there's a lot of effort, a lot of logistics in getting solar panels out to sites and things like that. Unfortunately, I don't do that. So I use other people's labor.
I had a question. So I was curious with the byes. I don't know my salts or my sodium cycle so well, but knowing that usually it's you have it in NaCl, which is very soluble. So you're working in a wet environment. You're applying, you've applied it with the wood or where you buried it, and it's also naturally there. Just curious how those interactions between the sodium and water redistribute it that might change how that bye is being used, or the wood, like the length of the effect of your wood treatment for decomposition.
Yeah, so it's a very good point, because these byes get flooded. Like, we only really go there in the dry season because you can't access them in the wet season. They're completely underwater. But that would definitely move the sodium around. And so we concentrated the efforts in particular sites. But the idea or the hope is that it becomes soluble and move around the bye, which I think it does, because there are definitely sites of intense mining by elephants and things like this at particular places in the bye. But these are kind of spread around. And so I think the sodium does get spread around quite a bit. In terms of the decomposition experiment, I think, I haven't thought about that all that much, but I think we soaked it in like salt water, essentially, to try and mimic that kind of effect, to think that that's probably how these animals are getting it. And whether the animals are eating the soil, there's a fair amount of geophagy in the byes, but they could also just drink the water and get it that way. And so I think it could affect the decomposition experiment in that way, and we're trying to mimic it as much as we can. So instead of adding salt to the wood, we're just soaking it in water, in a salt solution.
And another question from online asking if there's any intention to translate some of this work to U.S. wildlife.
I think, like, fundamentally, I don't really do much work in the US or any really, but the hope would be that these principles would be transferable, so we could think about things like bison in the same way that we think about wildebeest. And really, we're trying to understand the way that animals shape these ecosystems. That's kind of the work we want to get to, where we do these comparisons between Africa and other continents, is to think, how does North America function because we lost all these bison, for example, or as wolves come back, or as New England gets reforested. And so, we're trying to think about it quite at a fundamental, basic level to understand these impacts, to then apply it to particular examples. So we could think about how the prairie functions differently and how fire interacts with bison. Not that I know all that much about them, but I think so conceptually we can, but I haven't done any work in North America, but there are people doing similar things. Obviously, trophic cascades in Yellowstone is a famous example. But the idea is that these concepts, this understanding would translate anyway. Yeah.
Any other questions? All right, well, if you want to follow up, we'll be having lunch over in the lunchroom. And so let's thank Andrew and join him for lunch.
Thank you.